Introduce Dr. Ying Taur. He received his MD and MPH from New York Medical College and completed internal medicine residency at Long Island Jewish Medical Center. He then completed his infectious disease fellowship training at Memorial Sloan Kettering Cancer Center in New York City, after which he stayed on as an associate professor and physician scientist faculty for the last over 10 years. Dr. Taur's research involves the study of the intestinal microbiota and its impact on cancer patients. He's received support by the National Institute of Health and the MSKCC Center for Microbes, Inflammation, and Cancer for work specifically relating to the role of the intestinal microbiota in infections in immunocompromised individuals, particularly in recipients of allogeneic bone marrow transplant. He has overseen collection of over 18,000 fecal biospecimens from cancer patients and is principal investigator in a randomized trial of fecal microbiota transplantation in stem cell transplant recipients for prevention of transplant-related complications. Welcome, Dr. Taur.
Oh, thank you, Liz. I'm off mute, right? Okay, yes. Hopefully everyone can hear me. So yeah, thank you for that intro. It's a pleasure to talk to you. I'm hoping to give an on-target description of some of our work as it pertains to our clinical studies of populations in the microbiome, regarding the microbiome. The focus, it's something we do a lot of at Sloan Kettering, is allogeneic hematopoietic stem cell transplantation or bone marrow transplant. Not to oversimplify, but this is a situation where it's an extremely intense treatment. You're subjecting them to chemotherapy and radiation at very, very high levels, actually otherwise would be life-threatening. So rendering the patients in a very susceptible state. The transplanter takes ownership of the damage and the danger that they put the patients in by giving them stem cells. This is how it's doable. You give them the chemotherapy, radiation, the conditioning, and then they sort of save their life with hematopoietic stem cells. And that ends up being their new immune system in the allogeneic case. All the while, because of their susceptibility, they're subjected to a lot of antibiotics. We thought this was a really interesting population to look at because if you take care of these patients, you know that they have a lot of gut-related complications. They have a lot of infections, but then also other types of complications that really make bone marrow transplant a challenging population and making them some of the most complex patients you've seen. And more specifically, if you look in the literature and you talk to people who have been around the block in this field, they'll agree that there always was this sense that the gut is very, very intricately involved in terms of what is going on, and it's the link to complications. And sort of the microbiome, was very interesting to us because, let's see what it can explain in terms of what these patients go through. This led to a sort of biospecimen protocol, which we started over 10 years ago, where we're collecting samples as these patients are getting transplants. So I'm just showing you snapshots of the process where we're collecting and then we're freezing it down for storage. And then when we're ready to analyze, we extract the DNA, specifically the bacterial DNA, or viral or fungal. And we sequence that and then we analyze that on as best we can and try to provide a picture of the microbiome, essentially profiling the composition of their gut at various time points. because it's been going on for 10 years, this is just showing you how many samples we've collected as of yesterday. I just updated this plot because because I don't think too many people have collected more stools than I have. So here's what do bone marrow transplant patient guts look like? Well, here's a phylogenetic tree of the bacteria that showed up in all of those samples, almost all of those samples that you saw in the previous slide. And this is just a phylogenetic tree showing the various groups of bacteria. In ID, I'm an ID physician as well. Obviously, we do not talk about bacteria in terms of their phylum, but you have the Bacteroidetes, which are these obligate anaerobic gram-negatives that seem pretty central to the gut. You also have on the top this Firmicutes over here, this big clade, probably in most people the largest phylum, contains on the one hand obligate gram-positive anaerobes, which I usually call collectively the Clostridia, but then also a bunch of aerobic bad guys, I'll say, such as Enterococcus, Staph aureus, Streptococcus, which we certainly spend plenty of time treating. And so you might say the phylum is a little bit, has a split personality. So the phylum is a little bit too big. It's hard to describe that group of bacteria. You have the actinobacteria at the bottom. These are these gram-positive rods. And if you've heard of actinomyces, of course, but there's also other things like Corynebacterium. It also includes weird things like nocardia and Mycobacterium tuberculosis. So another sort of diverse phylum. And then Proteobacteria, the red. Think of every gram-negative you've likely treated, like E. coli, Klebsiella, Stenotrophomonas, Pseudomonas. These are all under the Proteobacteria umbrella. And it's worth noting, so yes, fine, all of these things are from the gut, but it's important to note that out of all those things I said, if you look at the top half, the Bacteroidetes, and one half of the Firmicutes, the Clostridia, that's actually 99% of the normal gut flora under normal circumstances. In other words, the stuff that we spend all day treating, like E. coli, the gram-negative such as E. coli, Klebsiella, Pseudomonas, and the gram-positive such as Enterococcus, Staph, Strep. I mean, that should be less than 1% of the gut flora at any, under normal circumstances. Normally your gut is filled with these obligate anaerobes that you've never heard of. And you've never heard of them because either A, they don't grow in culture at all, or B, they just don't come up in a natural conversation when considering infections. They're fairly innocuous and not really implicated in any sort of infection that you've ever heard of. So, so then so that already is an interesting question. So looking at BMT, what happens? This is an early attempt at looking at the diversity of the bacteria as a patient goes through transplant. And what you're seeing is a summary of the experience of 94 patients where we collected stool samples in those patients as they went through their transplant. Day of transplant on the x-axis just means day relative to the actual transplantation, the transplantation being like the administration of stem cells, where the minus period is where we were giving them the chemotherapy. And then afterwards we stopped and we're managing their neutropenia. The y-axis is the diversity, which is just a calculation of like how diverse is their microbiome at that time. And the dots are just a single stool from a single patient, and there are 439 of them in this picture. So summarizing that experience, you can see that looking at the average line, the sort of smoothed average line, there's an immediate plummet in diversity as soon as they undergo the process. But what we could also see is that there was a range of experiences, from way below this line to way above. In other words, like people who plummeted to 0 diversity and stayed there, to people who had a microbiome that didn't actually seem to care about transplant and for some reason remained diverse throughout the process. Here's a single patient, which I think is a reasonable intro to what is happening in these patients. So at the top is medication administration. You can see we're giving the conditioning regimen, the fludarabine and busulfan, and we're giving supportive antibiotics. In the middle is white blood cell count. You can see that the conditioning regimen gives them the neutropenia, and then later it's detectable again, and that's the new graft. But down below is a vertical bar representing a stool sample that we collected from the patient. Now, I'm not showing the legend, but the colors represent like a single species in the in that person's gut. And I'll just say that like the aqua, the pink, the brown, the sort of olive green, all represent healthy members of the microbiome that you typically would see in someone who is, not getting antibiotics. And this patient had that to begin with, but then you can see soon after the treatment, it becomes abnormal. And in this case, a minority member, Enterococcus faecium, expands and actually ends up taking like 99% of the space. And that kind of persists for as long as we measured, at least in this patient. So panning back to, what kind of patient experiences did we see? We saw all kinds of things, and I'm just trying to show that here. We saw people that were dominated. This is the previous slide where a single bug expands and sort of takes over the gut space, where the bug can vary between Enterococcus faecium to, Strep salivarius to Klebsiella pneumonia. While the organism can vary, the expansion is always some sort of minority member coming to the forefront. We saw resilient, where, they seem to get disrupted, but then sort of quickly snapped back, and to a state that's resembling something similar to what they walked in with. And then to unfazed. The unfazed is like these people appear to have a bulletproof microbiome. It's as if their gut did not care, that antibiotics and chemo were being given. These were very, very rare, but once in a while we saw that. My question is, who are these guys and what did they eat and what did they do right? That's something I don't quite have the answer to yet, but is obviously, a subject of great interest. In terms of the relevance, an early and relatively simple endeavor was to take the sample collected at the time of engraftment, where we can detect that new graft when the white cells are coming back from neutropenia. And we simply asked, well, does that tell us what happens to the patient? And what we found here was that if your diversity was low, which is the red line at the bottom, then you were more likely to die of any cause. That's on the left, overall survival. And, and more specifically, you were more likely to die of a transplant-related death, which is on the right side. And we sort of calculated hazard ratios for that. If you, now, I think your immediate question might be, well, hey, wait a minute, like, not so fast. Couldn't this be confounded by anything? I mean, like, how do you know that they're not dying simply because they're already sick and they got antibiotics and they messed up the microbiome, but it's not causal, you know? And I think that's a reasonable argument, but I will just come back with at least this is, you know, in this study, when we try to adjust for known markers, predictors of mortality in this population, such as those listed below, and including antibiotic administration, you know, this association was extremely strong. I mean, it was, I would almost say it was like, it was very difficult to get rid of the signal and adjust it away due to our known factors. Okay, so now I want to talk about a few complications that I think can be telling. And one of my favorite subjects is fever and neutropenia. Here I'm just showing some of the members that, some of the bloodstream isolates that you see when you're treating someone with fever neutropenia. And I'll put it to you this way, you know, when I was a fellow, one of my mentors, Kent Sepkowitz, he told me, just think of fever neutropenia as like, I shot you in the gut with a little gun, and now your gut is full of holes, and now gut bugs can just go into the bloodstream, and our job is to treat the bugs that, you know, go into the bloodstream until they recover their counts. And that was very helpful for me at that stage. That, you know, helped me understand, because I didn't, honestly, the literature does not clearly tell you that the bugs come from the gut in many instances. So that was very helpful to hear. But I had a lot of questions about this. I mean, if we know that the normal gut typically is 99% anaerobic, then why is this list filled with aerobes? You know, like, why is it always frequently like, you know, just those minorities that I spoke of? And, you know, how can we just never ever see, you know, something else? And just to sort of drive that point home a little bit further, here's some additional data where I'm just putting next to it a pull of all the bloodstream isolates that we see during surgical peritonitis, you know, a patient population we also have. And the most immediate thing I would point out is that this list has anaerobes, even ones that are hard to grow. And, you know, and another difference I would say is if you look at the Enterococcus, much more resistant, like in the fever neutropenia group than in the surgical peritonitis group. And I think that this is, you know, I think that this actually is hinting at a fundamental difference between these two types of gut translocation. One is, you know, one is a subset of the bowel flora, and the other one is less that. And so this was something that I think is worth knowing in order to think properly about fever neutropenia, which I think I have always found, you know, confusing. So here, sure enough, is what we found in people who had positive bloodstream infections, is that we saw that domination with that organism occurred. And so here's the very first one we found. Here's a patient who came in with a normal flora, and then very soon afterwards was overrun with VRE. 4 days after that, persistent bloodstream infection that continued despite a lot of attempts at treatment until 2.5 weeks later when she was dead. You can see us, you know, you can see us trying to administer everything under the sun in a sort of desperate attempt to treat this thing, but to no avail. And so that was, you know, that was like a eureka moment. And then subsequently, we have continued to see the phenomenon. And here's just a smattering of some of them. Again, the pattern being that, you know, the thing that expands eventually ends up being the bloodstream infection. So here again is the VRE up top, you know, upper right. Here's an example of E. coli, and then Klebsiella pneumonia. The fourth one is an interesting example of E. coli that was ESBL producing, followed by VRE, and you can see them both sort of making themselves known in the microbiome. And then other things such as Enterobacter cloacae, Stenotrophomonas multifila. Another interesting one at the end is Klebsiella pneumonia and then Staph epidermidis. So if you're wondering whether Staph epi is meaningful during fever neutropenia, well, the answer is it can be. It can actually be, rather than be a contaminant from the skin, it actually about half the time is in fact a gut translocation. Really, really interesting. I did not, I was not keen on that fact until we came across it. Here's another interesting angle for this. Here's a patient who I'm showing the temperature in the middle, and you can see they have a fever, and then we check blood cultures and the cultures are negative. All right, and of course we start our fever neutropenia treatment, as you can see up top. And what ends up, what the data actually shows is that the person has domination with something called Olsenella uli. Certainly I've never heard of that. If anyone's heard of that, I'm very impressed at your ID knowledge. But but this is an actinobacteria that I don't know anything about. And I just put this slide up here, not because I want to teach you about Olsenella uli, but just to say, hey, remember that 80% of fever neutropenias end in culture negativity. Like you try to do it and then you get negative cultures. And a fun question is like, what is happening in the guts of those patients? And the answer, among other things, is, for example, this thing. But then there are all sorts of other things that rise that I had never heard of. And fascinating question is like, oh wow, like, okay, so even the ones that are culture negative, those people are also getting dominated by something. And so like that just helps to understand like the process a little bit better. Okay, so what drives this thing that I am calling domination? In other words, the expansion of the minority. And one answer to that is antibiotics. But the thing I want to impress on you is that not all antibiotics are equal. Here is a patient who is, has a nice population of pinks, which are Lachnospiraceae. They're, it's associated with good with good flora, and the person gets various antibiotics, but it's not until the patient got metronidazole that the gut like goes south and is overrun, like in previous patients with Enterococcus faecium. And I would have called the previous antibiotics like broad spectrum, but as you can see, they actually don't do much to the microbiome. And so that's very, very telling. And I think turning AMP on its side in many ways is, we say narrow spectrum, broad spectrum, but what do we think the thing is doing to the microbiome? That might be a very important question, especially when we're talking about selection of MDROs or C. diff. And so that's something that ought to be kept in mind. If we looked specifically at the risk of VRE domination, we found that administration with metronidazole was associated with over 3 times more likely risk as compared to IV vancomycin, which had no impact. This is, the point is VRE is a very misleading name because IV vanco actually does not select for VRE. That actually, I'm not the only person saying that. Even studies from 20 years ago show that administration with intravenous vancomycin does not select for it. And instead, antibiotics with potent anti-anaerobic activity are the drugs that select for VRE. Those are the top selectors of this bug. This is just a nerdy paper. Liz will tell you I'm like one of the biggest nerds at MSK. And so this is a Bayesian regression where we took a handful of patients and collected stools intensely on them. And we took their antibiotic administration. And what we did was we got an independent readout as measured by this posterior distribution you're looking at of the impact of each antibiotic. And so the more negative this distribution goes, the more impact it has on the obligate anaerobes. And here we have metronidazole, meropenem, piptazo, cefepime, vanco, cephalosporins, fluoroquinolone, trimethoprim, sulfamethoxazole. So now, you guys are ID people too, right? For the most part. Isn't it true? Like, I don't know about you, but like if you were to just give me these drugs and ask me to rank them in order of anaerobic activity, I probably would have put it in this order, more or less, something like that. And so, yeah, that's what we found is that perhaps not surprisingly, the drugs that we know have potent anti-anaerobic activity are the most disruptive of the microbiome. On the right, I'm just showing you the range of experiences that we had in our data. And so, yeah, like, I think that, I take this as like, that makes me focus more on the anti-anaerobic activity of the drug when I think about like, when I think about what the thing is doing to the flora. Now, I'm going to pivot now to another antibiotic that I just want to talk about, and that's oral vancomycin. And this, we had an experience where what we were seeing was that there's a lot of C. diff in our transplant patients, and what we were finding was that the primary teams were giving longer and longer courses of vancomycin. I'm going to admit, even some of our own infectious disease attendings were doing this practice of extending the oral vancomycin while they were on other antibiotics such as piptazo, where the idea was I'm preventing some C. diff because I know this person had, usually the patient had recurrent C. diff or something like that, and the idea was let me put them on oral vanco so that I can prevent the C. diff from happening every time they get the insult of other antibiotics or something like that. In other cases, they actually did prophylaxis. This was definitely a practice that was being done in the transplant, in our transplant patients where, they just didn't want to hear from C. diff, and so they just kept them on oral vanco as a prophylaxis. The graph is just showing you like to what degree we saw it. I mean, we saw it in a ton of cases, like something like up to 10% of administrations. But which ended up being like 1.5% of patients. So these patients getting a ton of repeated prolonged courses. And here's the thing that I think is not well known, is that oral vancomycin is actually like one of the most devastating drugs to the microbiome in existence. It's even worse than metronidazole, which was at the top of my analysis in the paper we showed before. I'll just point at, okay, so first of all, like in the top left, I'm showing a single patient, just showing you what vanco, just another visual of what the oral vanco does. But then also, if you look at this, the snapshot below in the lower left by Johnson and Girding, that was a study actually in the 1990s, very, very long ago, where they tried to treat people who were colonized with C. diff. And what they found was that C. diff carriage and infection increased in anyone who got oral vancomycin. Their conclusion then was that although treatment with oral vancomycin is temporarily effective, it is associated with significantly higher rates of C. diff carriage 2 months after treatment and is not recommended. That is supported again by this microbiome study on the right, which was done by Eric Paymer, who's now at University of Chicago, but also was from Memorial Sloan Kettering. And he showed by looking at mice that got oral vancomycin administration, that oral vancomycin ended up killing gram-positives, but also gram-negatives, and was much more disruptive than metronidazole. As you can, I'm trying to show one of the pictures that indicates that, where you can see, like, you know, the C and D are the, you know, are mice that got the oral vanco, and you can see there's a visually there's much more disruption there. Their conclusion was that oral vanco does lead to prolonged susceptibility to C. diff and selection of MDROs. So, and then just to sort of like try to change the practice at our institution, we did this small data poll where, you know, we said, like, you know, yeah, like oral vancomycin looks like it's amazing because when it's on, you do not have C. diff. But the key question then is, if there's a paradoxical increase in CDI risk, then the real way to measure the effect of oral vancomycin is to check not only the CDI risk while you are on the oral vancomycin, but afterward. And according to the, you know, to the Girding study, they would say at least 3 months look for that CDI risk. And if you do that, then we, you know, at our center, we got the following. Down below, we're looking at 5 years of MSK data. And if you have just no history of CDI and you're an inpatient and you just get antibiotics, then your risk of C. diff overall was 3%. If you had a history of CDI, like any past history of CDI, then that rose to 10%. And here's the kicker: if the situation was you have a history of CDI, you got antibiotics, and someone said, I'm gonna give you an extended dose of oral vanco, they actually had slightly higher rates of C. diff than if they had than if you just left it well enough alone and just, you know, given the antibiotic. And then, you know, obviously if you have C. diff, then you'll treat it, but to not treat it, to not automatically prophylax against it, you know, at least with this small data pool, we saw that actually, like, there were higher rates. And I think the point is, you know, just, you don't want to assume that this drug is doing great things, you know, because I think that because I don't think enough consideration is always given to, like, how does the oral vancomycin leave you, and what sort of implications would that disruption have? Okay, let me. Again, looking at selection, this was an angle that I really find fascinating. And this is looking at the presence of antibiotic resistance genes. And what you're seeing here is one of the patients where below you can see that, you know, there is a patient going through bone marrow transplant, and they undergo through like these shifts. This person had an ESBL bloodstream infection, as, you know, matched by the rise in E. coli, this red thing at the bottom. And what's interesting is to see the flux of antibiotic resistance genes that occurred during these transition states. And so, I mean, some of this is complicated, but I'll draw your attention to, okay, this right up top, this pink thing, you see this like rise of CTX-M155. Well, that's a ESBL gene. And so that correlates very nicely. And then just a side point, you see this, and this is very quick, but we have several views of this now. You can see that while this patient was getting treated, it's worth noting that they got cefepime before the ESBL rose. And, you know, I think we know that cephalosporins are pretty good at selecting for ESBL. But also note that there's a temporary rise in this aqua, which is not labeled, but is a Bacteroidetes. And the thing to know about Bacteroidetes is that they are actually obligate anaerobes that do have a resistance element, and that's CFX-A6, which is over here. So in other words, in other words, in this patient, what's happened is cefepime is given, and then that's like saying, and then the two things that rise are E. coli and Bacteroidetes. And it's as if, we're saying, hey, is anyone resistant to this? Please raise your hand, there's a rise in the resistance gene flux, and those bugs, come to the top and are the most notable elements in the microbiome during that time of selection. These are fast-moving pictures, and we'll need more data to know more about it, but it's stuff that I think is very telling and interesting. I'm just going to spend, 2 or 3 slides talking about the immune system. Is that we now have a lot of data that makes it it's no longer controversial that there is a connection between the microbiome and the immune system. It's just been shown many, many ways. The way it impacts in the cancer field is several ways. One of the ways is this science paper that came out in 2018, where we're using immunotherapy, specifically PD-1 blockade. And what they found is that if you took the stool of patients who had responded to immunotherapy and you put them in mice with tumors, then the tumors of the mice responded. Whereas if you gave them stool from a human who did not respond to PD-1 blockade, those tumors were not well. There was no response, and this was a great thing where 3 separate groups had shown the same thing, and that was published all together in Science. So it was really cool to see very resounding evidence for that connection between the microbiome and the immune system. Here's another study. It's only 4 patients, but this was a study that came from Japan, where these are transplant patients who are being treated with third-party FMT for graft versus host disease, and so the point is, all of these patients got FMT, as you can see by the red arrow, and 3 out of 4 of them had a sort of night and day response to their diarrhea. So if you look down at diarrhea, you can see that, all of them, their diarrhea improved after getting either 1 or 2 FMTs. The only person that did not respond effectively was case 4. And I can't help but point out that that's the person who got dedicated anti-anaerobic, dedicated antibiotics against anaerobes. And maybe that's something that needs to be borne in mind. This is a study that we did, which was involved, but I'm just going to say we looked at butyrate producing bacteria, in other words, the anaerobes. And we saw that if you did not have a good composition of anaerobic butyrate producer bacteria, then you were more likely to get lower viral respiratory tract infection. So again, more evidence of this connection between microbiome and immunity. Probably, we would probably say the T-cell immunity if we had to be more specific. Okay, so bringing this stuff together, this is another phylogenetic tree where I'm trying to show some of the studies that have been done in bone marrow transplant. And you can kind of see a pattern rise here. The blue groups represent the bacterial groups that have been associated with positive outcomes, such as better outcomes in graft versus host disease, bloodstream infections, viral infections, mortality. And the red are the groups that have been associated with negative outcomes. And again, they just overlap in the same places. And so what are the good groups? The blue is 3 things. It's the Lactobacillus, and number 2, the Rhamnococcacea, and number 3, Bacteroidetes. Whereas the bad guys are repeatedly things in the bacilli group, which are gram positive. This is like staph, strep, enterococcus. and proteobacteria, which is, as we said, E. coli, Klebsiella, etc. Sometimes I call the three good ones the trifecta. And let me just show you a completely different study. This is not done by me. This is just an infant who was in a nice paper by Koenig. They took a single infant and just sequenced them every day for 2 years. And you can just see like what's going on. You can see that the infants started with anaerobic stuff in their gut. They're when they're born, they're virtually sterile, and you can see these successive changes over the 2 years until finally at the end, well, hey, look at that! It's actually the trifecta. It's Bacteroidetes, Lacno, Romano. So this, at the end on the far right, is what you want in your gut. That's what you want for stability, and that's what the gut expects and wants. And in order for good, good health. And so, I think that the results in the bone marrow transplant field do match up with other microbiome studies in other fields. Let me just say final words. 2 more things, I guess. One is on probiotics. Well, can we do anything with probiotics? Do those ever work? Isn't it true that I think many of us don't know what to think about them. But I would say one thing that some data has come out that is kind of a strike for them. And this, for example, this study where they looked at mice and humans where they got antibiotics, and they would do one of 3 things. They would either do nothing, or they would give them probiotics, or they would give them an autologous FMT. Autologous meaning the FMT actually came from themselves. And what they found was, interestingly, the probiotics were the worst group, like even worse than doing nothing. And so their conclusion was that probiotics actually perturb rather than aid in microbiota recovery back to baseline after antibiotic treatment. There are actually 2 other studies in 2018 where they were negative for probiotics. So 2018 was a very bad year for probiotics in general. So what's going on? So I'm showing the family tree again. And I'm saying that and I'm also showing the good members of the microbiome. Well, so where are probiotics? In the green is probiotics. We have Lactobacillus, species, which are all in the red zone. And we have bifidobacteria, which are sort of these ones out in the white area on the lower left. And so this is, I think for those of us who study the microbiome, it's actually really clear why probiotics don't work. I mean, probiotics are the right idea, but they're totally the wrong bugs. This is not what your gut is filled with and not what your gut expects. And so it's not surprising that, the data for them has always been extremely plus minus. So now just final, rounding the last corner here. I've shown you that in that infant, we have something called primary succession. This is like the infant slowly growing from nothing to like full forest at the end of 2 years. So if that's primary succession, secondary succession is what happens in transplant patients. That's like you had a forest, but then I burnt it to the ground. And now you have to grow it back naturally again, sorry. And so thinking about that way, like if it is true that, transplant patients really need that in order to resist MDROs, and they need it in order for their immune system to be intact in that, at-risk period, then how do we do something about that? So this led to the clinical trial that we did, where we said, like, should we take ownership of the microbiota that we had destroyed? And the study was to enroll patients at the start of their transplant. We would collect their healthy feces. We'd actually even confirm that it was healthy by sequencing it and just making sure it looked okay. Putting that in a freezer and then letting them go to transplant, where they're going to World War III. And then when they were done, then we would randomize them to treatment or control, where treatment was we take that stored feces out of the freezer, thaw it down, and then give it to them via retention enema. And then control was just standard transplant care with no modification. And in this study, we were looking for a few endpoints: C. diff, viral infection, bacterial infections, graft versus host disease, and TRM. Down below, I'm just showing you a picture of the stored stool. We put it in bags that are basically the same as the bag that we use to store the actual stem cell blood. So the study is actually technically on, so we're close to accrual, but it's, but we haven't officially closed it yet. But here's just a snapshot of what, here's one patient just to show you what happened. So again, you can see the disruption that we cause. The red stool is the one that we stored and froze down, and then I'm with the dotted line. We're trying to show that this person randomized to auto FMT and got it back, and you can sort of see that it sort of looks at least closer to what that patient originally had, and importantly, it maintained itself. It stayed consistent despite a few insults with antibiotics. And then, if you're one of the nerds who actually cared about the antibiotic resistance genes, here's a view of what happened to the resistance genes during the transplant. So you can sort of see, yeah, of course the resistance genes rise during the aberrant period, but then as we do that auto FMT, the resistance genes also look better. Okay, and so that's it. I just want to remind you of a time where we thought the best way to do transplant was to put the patient in a sterile room. And here's a picture from way back showing you that. So the patient would be put in this sterile area, and you actually had to have a wall of HEPA filters in order to keep the place sterile. In order to do this sort of a thing, you would keep them in here and then repeatedly culture the gut of the patient, and if it grew anything, you would give them antibiotics that would treat that. And if loved ones ever wanted to visit, they had to be on this side of it, and if they wanted to hold hands or something, they had to stick their hand through this barrier with the two circles. And I just love looking at this and in an incredulous way, and just because I'm the guy who wants to throw stool all over the place, so it really is very a huge contrast to see the range of thinking in this field. Just as a point of interest, the previous head of infectious disease at Memorial Sloan Kettering was Don Armstrong. He was friends with the prior head of ID at MD Anderson, and though they were friends, they disagreed forcefully on the point of whether or not protected isolation, i.e. the sterile room, was a good thing or a bad thing. Don Armstrong was of the opinion that it was not useful, it was too expensive. Jerry Bodie was, who I think is a forefather of fever neutropenia care. In this case, though, was a holdout for protected environments, and they actually had a published paper duel. This is something we don't do anymore. They dueled each other by writing, opposing papers, and these papers are really, really fun to read. But I just thought I would, if anyone else is interested, read these papers. Very, very interesting, to do this, because unfortunately we don't do this anymore. But yeah, so that's everything I had to say. I thank you for your attention. This was the colossal effort of many people. I'm trying to list here, and so I'll leave it at that. Thank you. If there's any questions, I'm happy to discuss.
That was a great talk. Thanks so much. I just had one question. You know, it's not terribly surprising to me that Piovanco disrupts or sort of destroys the balance of the microbiome, particularly in transplant patients, as you're showing. But why is it so much greater even than metronidazole? Is it because oral vanco isn't absorbed and you get the concentrations are just so high intraluminally? I mean, it seems to me, I know there's plenty of gram-positive anaerobes in the gut, but it seems like there'd be even more gram-negative anaerobes. And just any thoughts on your, I mean, it just was a little surprising to me that Piovanco is even more of a nuclear bomb than metronidazole.
Yeah, that's a good question. I do think that a lot of it has to do with pharmacodynamics, right? You remember how, I don't want to assume, but, you know, do you remember back when like we were using C. diff treatment, either metronidazole or oral vanco, and then became clear that oral vanco should be the better treatment. Mm-hmm. And there was all this concern, for example, for metronidazole, does metronidazole touch every single part of the gut? And there were these studies that seemed to show that metronidazole be absorbed, like all of it, 100% of it would be absorbed systemically and then excreted later in the gut. And so the concern from the C. diff perspective was we're not treating 100% of the gut, right? And maybe that explains why its effectiveness is lackluster after looking at tons of data compared to oral vanco. Oral vanco, meanwhile, does not get it systemically absorbed at all, marches like a tank through the gut lumen, destroying everything in its way. And I think we get that sense when we look at, you know, the pictures of what's going on as the patient is getting that drug, which, you know, meanwhile, IV vanco, I already said, like, it actually barely gets into the gut. Right. And that is why it doesn't select for VRE, probably. You know, there are studies that show, you know, you have to give IV vanco for something like 3 weeks before you start to see much of it in the gut, you know, and so I think that, you know, that's something I think it would be, I think that's the explanation. I don't know otherwise, because yeah, it's the same substance intravenously versus PO. So I think it's got to be
gut concentration. Yeah, I would think.
Yeah,
that's cool. Well, it was a really excellent talk. Thank you so much.
Pleasure.
Yeah, if there's extra time. So one, thanks to Ying for an awesome talk, and also to my ID colleagues for letting me crash their meeting. I'm a gastroenterologist, and Ying, I know your work through meeting Eric Paymer, et cetera. So I do fecal transplants, those sorts of things. Was curious, similar line, do you have any data about the risk of, or have you looked at the gut microbiome in fidaxomicin treated patients, especially with, you know, the push to use more vanc now and this issue that we have with, you know, more and more recurrent C. diff?
Yeah, that's a good question. So the answer is no, we don't have a lot of experience. We don't have a lot of clinical experience with FIDAX, and so therefore I don't have a lot of research experience with it. You know, the problem being that it's too expensive for anyone to seriously use. I had one. Its claim to fame is that it spares bacteroidetes, and I think that is a potentially good thing. That to me is a rational explanation for why FIDAX is associated with less recurrences, but it's not. But I feel like that drug has not stood the test of, you know, people using it frequently and experiencing it to know if that's actually true, because of the because of its expense. I myself used it one time and it failed. You know, like I had this person who I'm going clinical because I don't have the research to tell you, but I mean, I had this one patient who kept getting recurrent C. diff. He was a transplant patient. And every time after, you know, we would do like ridiculously long oral vanco tapers. And then as soon as he'd complete, he would just have it again. He didn't get an FMT because he was very stubborn and he thought it was gross. And so he never did that, which is unfortunate. So my idea was, you know, let me give him fidaxomicin. Like we tapered him and then we switched him from oral vanco to fidax. And I said, like, just stay on that for a month. And my whole idea was that's a targeted antibiotic. I'm hopefully just suppressing C. diff. And I was like, maybe for the month, he'll grow like a nice flora around it. And then I'll let go of the fidax and he'll be fine, right? But no, that didn't happen. He was on fidax for a month. He was on no other antibiotic, and then when he stopped the fidax, he got a C. diff again. So that's the only experience I can speak of, and I feel like anyone who's even seen the drug has stories like that, but no routine experience, you know, from it. I would love to. I mean, if anyone has routine experience with fidax, I would love to, you know, just pick your brain about it but I just feel like we don't. I can't get enough people in the room to talk about like what the drug actually does, short of what the studies are showing.
Thank you.
And any other questions? Dr. Taur, thank you so much. That was a wonderful talk. We really appreciate your time.
Oh, yeah, my pleasure. Thanks for
And
having me.
thanks, Liz, for bringing them our way.
Thanks for coming.
Thanks, Liz.
Bye.